Okay, can you tell me what genetic engineering is?
Genetic engineering is when you take DNA from another organism and you insert it to a vector.
Okay. That's the process. Tell me what it is. What is genetic engineering? What do we do with it? Why do we have it? Why do we use it? Go ahead, Kyle.
It involves building recombinant DNA made from 2 or more different organisms.
Okay, give me an example of why we would use genetic engineering or why we need it.
To make, for example, if you could insert a gene into a plant that could give it resistance to insects or has more vitamins.
Okay, very good. Okay, so why don't you use your model and take me through the steps of genetic engineering?
So you have
This is the plasmid, aka the vector. And inside here we have, these are the genes, this is the DNA we cut from the organism.
What do you cut it with, Kyle?
enzyme.
Your EcoR1, okay.
Yeah.
The green represents the gene of interest, which we want to put inside it. And this blue is very important too, because that's tetracycle, it's the insulin gene, tetracycline resistance. So that will be a major part too.
Okay, so show me how it works. Show me what happens.
Well, first of all, this is stuck together originally. Then we cut it with this EcoR1. We cut this too, except for we cut it twice. This only once because we're only inserting it with 2 sticky ends. And since the sticky ends match up, we can then insert it into the vector and make recombinant DNA.
Well, we're not going to have our DNA in there, right? Because we cut it out. Right? The blue and the yellow and the purple. So take that out. Okay, so now we're left with that circular thing. Kenny, what's that called that he's got right there?
That
or
is
Kyle?
called the recombinant DNA.
Good. Okay, so now what do we do with the recombinant DNA?
Oh, we insert it into bacteria so that it can be cloned, because it's cloned naturally when it's inserted when the bacteria reproduces, which is the 3rd step in genetic engineering.
Kyle, what's the last step in genetic engineering?
Screening.
Okay, so now that I have this recombinant DNA in my bacteria, how do I know that it works? How do I know that I did it right?
Because it would replicate.
Okay, good.
Now, he knows that he did it right because what I told you is it has the tetracycline resistance gene, and when you add tetracycline into it, it will kill off all the other bacteria except for the ones who have the tetracycline resistance in it.
Nice.